if we were at a very low pH, we would have to write the N-terminus like this. But even if we were at a low pH, we would never write this nitrogen like this. This would always be wrong because this is an amide nitrogen. Amide nitrogens don't protonate. So what I have on the board here is wrong, so I'll erase that. This is not basic, it's not going to get protonated. Only the N-terminus nitrogen over here can get protonated because it's still an amine. And by the same token, what type of functional group is this?
Carboxylic acid.
So can that change forms?
It can.
Yeah, absolutely. Now here we have a carboxy carbon, but it's no longer a carboxylic acid. So again, we don't need to worry about this taking two different forms. This is obvious that it can't take two different forms because it's lost the whole acidic proton over here. So all of these amino acids have these carbonyl carbons, but the only one that can take two different forms is the one at the C-terminus. All the other ones can only take one form because they're buried inside the amide bonds. Once you're buried inside an amide bond, you can only take one form. Let's go through this tripeptide then and identify all the acidic or basic functionalities. So we'll need our table again. We'll start with glycine. Is the main chain nitrogen here? Something that can be go through acid-base reactions. For glycine, do we need to worry about the acid-base properties of the nitrogen?
Yes.
Yes. So what's its pKa?
Lysine is 9.6.
So I'll write down that 9.6.
So I have a question. If our side chain is neutral, then we don't have to worry about the pH affecting it?
If it's neutral, you mean if it's neither acidic nor basic?
Right.
That's right. If side chain is neither acidic nor basic, then we don't need to worry about pH affecting it. That's right. You know, this will be more interesting if we change this from serine to tyrosine. Let's change that last amino acid to tyrosine. Glycine, we have to write down this pKa. Now, does glycine have an acidic or basic side chain?
No.
No, because there's a dash in the side chain column. Do we need to worry about the pKa, the acid-base properties of the carboxy side of glycine?
No.
No, because that's buried in the amide bond. So even though in the table it says that the carboxy side for glycine has a pKa of 2.3, there's no point writing that down because that's not really that's not a actual issue when this is buried in the amide. Now let's move on to alanine. Do we need to worry about the pKa of the alanine nitrogen?
No.
Because that's part of an amide. Do we need to worry about the pKa of the alanine side chain?
And it's neutral, so no.
Right. Technically, we probably shouldn't use the word neutral. Neutral refers to solutions. It doesn't refer to molecules. We would simply say it's neither acidic nor basic. But that's the right basic idea. Do we need to worry about the pKa of the alanine carboxy carbon?
No.
So we didn't have to worry about any pKa's for alanine. Do we need to worry about the pKa of the tyrosine nitrogen?
No.
No, because it's part of an amide. Do we need to worry about the pKa of the tyrosine side chain?
Yes.
Yes. So that is,
It's acidic.
that's right. It's acidic, that's right. And what's its pKa?
10.1.
So we should write that down. And do we need to worry about the pKa of the tyrosine carboxy carbon?
Yes, we do.
Because this really is a carboxy group here. It's not part of an amide.
Right.
Good. And what's its pKa?
That is 2.2.
2.2. Right. You can see it's important to be careful about getting the right numbers. 2.2. Okay, well let's find the PI for this tripeptide.
Do we need a pH first, or we don't?
Now remember that a few minutes ago we did a problem about PI. You need to go back in your memory. What was our method that we had to do?
With a really, really low pH.
Right. Not because the pH really is low, but just as a thought process.
Okay.
Just as a thought process, it helps. So we're going to have to do a lot of work before we can do the PI. There's a whole set of numbers we're going to have to develop before we can find the PI. And the first thought step is to imagine that we're at a very low pH.
Already protonated. This is also already protonated, so we'll have +1. Wait, let's see. So if we gradually increase, we start deprotonating. This will be deprotonated. So we have plus minus 2.
Let me give you some help with those steps. We started by imagining that we were at the very lowest possible pH, and that would give us a +1 charge. It's good that you saw that even when this is fully protonated, it doesn't have a +1 charge. Because remember, and we can see from our table, there's no little C here. So this is acidic, not basic. No, that's not the mistake that you made.
Yeah, I see it now.
Oh, okay. So this was good. So you're right that fully protonated is +1. So then what do you think went wrong here?
This wouldn't have a minus charge, it would be neutral.
Okay.
So we would have a -1.
Still a problem with that. This is the picture that we would have when everyone's protonated. Now, the first threshold that we can cross is the 2.2 over here. Now, when we cross that threshold, who's gonna deprotonate?
The 2.2.
And what's the net charge that's going to give us?
A -1. No, it would not give us a -1. We'd be neutral.
Right.
Oh, so we'd have a 0.
It would have a 0 net charge. So far, we haven't done anything to this nitrogen.
Right.
When the pH crosses the 2.2 threshold, that's far away from the threshold for this nitrogen. All we've done is deprotonate this carboxy carbon on the right. All right?
So then when the pK passes 9.6,
Right?
that one is also going to deprotonate, and that would make that amine on the end neutral. Or there wouldn't be a charge.
Right.
So we have a -1. And lastly, the OH on the top would deprotonate once we hit 10.1. And that would give us a minus charge. So we'd have -2.
Right. Now what?
Our Zwitterions, we need the PI of the entire tripeptide.
That's true.
So our Zwitterion is this.
Right. Well, do you remember what's the definition of the PI?
Oh, that's pH at which we get the max concentration of 0. So that would be the average of 9.6 and 2.2.
That's right.